Okay, so I'll be demonstrating how a biologist might use our platform to analyze their single cell data without having to code. So they just have to load in their data set and open it up here, and we detect what it is. And this is a data set I worked with when I was in the lab, looking at lungs from mice infected with influenza. So the first thing the user might do is just populate some information about the data that they know, and it can help with analysis. If they don't, we will detect it and do our best shot. So first thing that happens is some of the normalization as well as the quality control, and then you get this data set visualization of the cells. And the first thing you might want to do is label the cell types. So we're going to ask it to label our cells, and then while it's being labeled, we can look at some of the quality control plots that are generated and just see if it looks good. The pre-processing went okay. And then we can come back and we see it was labeled, and then show me the labeled UMAP. Now we can see what it would look like, and now you see which cells correspond to which cluster. And if I want to see it a little bit cleaner, I can just edit the plot here and see that this is the different cell types that is found in that data set. And this makes sense to me because it was a whole long data set. There's a bunch of different cells that are popping up here. Now, this was collected through multiple data sets in time. Show me a UMAP colored based off time point in the metadata. So I can see which cells are coming from which time point. And here we have the colors based off of the time point, and the purple cells over here are showing up at day 21, and on day 3 you have some of these orange cells showing up. So that indicates some biology that's going on that we can look further into. Next thing that you might want to do is see how much inflammation is in this data set, right? I can use a gene set that we've curated on the side here and ask, hey, can you show me how much of this gene set is being expressed through a feature plot, and then show me a violin plot after you score each cell type cluster for this gene set. So this will let us visualize how much inflammation is going on and which cell types are causing the most inflammation. So we have the 2 violin plots and the feature plot, so you can see there's some signal in the top end here, mostly some here, not as clear to see, and we can adjust that easily if we go over to the customize and modify this. And we get to see that a little bit more cleaner. And then we get to see which cell types are being mostly expressed through the violin plot here. And then once I'm done analyzing, I can just export by saving for publication. And these are publication ready graphs. And then I can save here and pick up where I left off next time. And if I go to demo, I can see it's there, and these are some old data sets that I've analyzed before. So that is Organism.